The first half of this event will actually consist of a recording from last May. So please ignore the date that you'll see on the screen and pardon the Zoom screen format looking a little strange. After that video, then we'll have the live Q&A with the researchers who joined us today and return to a Zoom format that you are more used to. Welcome, everybody. This is our 7th Organization of Biological Field Stations, OBFS, live from the field event. My name is Kerry Wininger with Sonoma State University's Center for Environmental Inquiry. We encourage you to view past recordings and learn more at thevirtualfield.org. For those of you who are instructors, be sure to visit thevirtualfield.org to access supplementary materials for your students, such as an instructor guide for live from the field events with suggested assignments and an event resources document full of publications about the specific research topics and projects being discussed today. Very quickly, here are some guidelines for this webinar. Remember, all participants are muted and your video cameras are off. During the presentation, please use the chat button to communicate with us and each other, and remember to choose all panelists and attendees so that everyone can see your comments. During the presentation and the live Q&A session with researchers, please submit your questions using the Q&A button, not the chat. If you're here as part of a college class, please type the word student in front of your question so we can prioritize it. And it's important to make sure your full name is visible, either as your Zoom username or typed in after the word student, so you receive credit for attending. Also, welcome if you are watching us streaming live on Facebook. We'll try to get to your questions too. Without further ado, I will turn this over to Dr. Tasha, Comendant, Conservation Science Manager with Pepperwood Preserve. Tasha is conservation scientist with over 15 years experience developing methods, tools, and science-based solutions that inform natural resource management decisions. And we're very fortunate to have her moderating this event today.
Thanks, Kerry, and thank you all for joining live from the field. We welcome classes, faculty, and other participants from across the nation and around the world. This is the 7th Live from the Field event. In this series, we take you on virtual field trips to research sites around the country and the world. As you'll see today, we are visiting 3 biological field stations and marine laboratories, places where people from many backgrounds come together to study the environment. Live from the Field events are a project of the Virtual Field, an international coalition of over 50 field stations and marine laboratories. This National Science Foundation funded effort brings the field to you. Find out more about events like this and other virtual learning experiences for college students at thevirtualfield.org. Today, we're going to get a look into the work of 5 scientists doing applied research and fieldwork that addresses societal challenges. And by applied research, I mean that they work on questions and systems that try to solve specific problems and provide innovative solutions to issues that affect individuals, groups, or society at large. So we'll see examples of how humans can work with farmers, fishermen, industry to better understand scallop reproduction and manage those populations. And we can learn from nature's design and help build better buildings and shorelines. And lastly, we're going to look at how data, information, and building partnerships can make our communities more resilient to fires and floods. So I'm really excited and honored to get to moderate this session of Live from the Field and introduce you to some examples of how science and field research directly benefit people and communities. And I hope this panel stimulates your questions and ideas. Each researcher has prepared an 8-minute video describing their applied science research. After the videos, the researchers will be available to answer your questions live. I'd like to encourage everyone to post the questions in the Q&A as we go along. Students, remember to type student and your full name in front of your question. So the Hurricane Island Center for Science and Leadership is a 125-acre island in Penobscot Bay, 12 miles off the coast of Rockland, Maine. They integrate science, education, applied research, and leadership development through year-round educational programs and a seasonal, environmentally sustainable island community. Hurricane Island would like to acknowledge, honor, and make visible that these lands are the ancestral lands of Native people. And the researcher you'll hear from first is Phebe Jekylek, a PhD student at the University of Maine and the director of research at Hurricane Island. She's been conducting research there for 6 years and will be talking to you today about science industry partnerships and population dynamics and interactions of aquaculture and wild species. One of her favorite parts about field research is literally immersing herself in the work and sharing the hands-on experience with learners of all ages.
Hi, everybody. My name is Phoebe Jekylek. I am the Director of Research here at the Hurricane Island Center for Science and Leadership in Penobscot Bay, Maine. I carry a few degrees. I did my undergrad at Boston University. I have a dual master's in marine biology and marine policy from the University of Maine. And I am currently working on a PhD in ecology and environmental science at the University of Maine. So my discipline really is marine ecology, and I do a lot of fisheries and aquaculture related work. I'm very inspired by the people that live on and make their livings from the ocean. So I'm really interested in doing applied research that can contribute to our working waterfronts, contribute to our coastal communities, and inform work that we're doing here along the coast of Maine. What has led me to where I am today? It's been a long and winding road, and the road continues, and I'm not sure that it will ever stop. I hope it doesn't, because I've had a really great time so far, and I've been really privileged to do a lot of amazing things. My first field experiences were in college. I did the BU Marine program and the BU Tropical Ecology program, and those were my first experiences doing field work and really connecting what I was learning in a classroom or in a lab to what was actually happening in the world around me. And so that was really, really influential and really just helped me see myself doing this kind of work. It was field science that really drew me, and it was really being out in nature and making observations, working with people, being on fishing boats. It was those hands-on kind of immersive experiences that really led me to the kind of science that I do now, and that I'm privileged to continue doing. And I just get to work with so many amazing people in amazing places like Penobscot Bay, Maine. So I'm really very lucky. I also spent time on an island off the coast of California called called Catalina Island, and I worked at the Catalina Island Marine Institute for about 5 years. That is where I learned what it means to be an educator. I learned that I have a natural love for education, and that it really energizes me and inspires me. And that's the first time that I got to really practice what I learned through experiential science learning, and I really got to share that with people. So it was pretty amazing. It's where I learned a lot of hard skills too. I learned to drive a boat, I learned to scuba dive, I learned soft skills like communication, listening, doing a lot of listening, and I was really lucky to work with a lot of different people that would come out to the island for really hands-on learning experiences. So I'm pretty passionate about both education and science, and here at Hurricane, I'm lucky enough to be able to do both of those things while working with amazing people in our local communities. So I'm going to tell you a little bit more about what that looks like for us here at Hurricane. As I said, I work for the Hurricane Island Center for Science and Leadership. Our mission is to integrate science education, applied research, and leadership development through year-round educational programs and a seasonal, environmentally sustainable island community. May through October, we welcome middle and high school students, adult programs, colleges and universities, and visiting scientists to our island campus. This campus is also home base for the majority of our research efforts, of which I'll talk to you about today. We are located in the heart of lobster country in Penobscot Bay, Maine, and are part of the Fox Islands archipelago. The island itself lies about 12 miles off the coast of the mainland, so hurricane relies heavily on boats for all of our transportation and a lot of our work, and very little gets done without a lot of teamwork. So before I dive in more deeply about our research, I think it's really important to mention that there are very little research efforts, no matter where you are, that are done alone. The vast majority of research, and especially that happens on hurricane, needs a team to make it all possible. This is our team, myself, Madison, and Carl, heading out to the north end of the island to our 3.2-acre aquaculture farm where we grow scallops. Maine has a growing aquaculture industry along its coast, and scallops are one of the species being cultivated on farms. They're grown in different gear types, and we keep some of our scallops in cages that sit on the bottom, like the one you see being pulled up here. Our research focuses on evaluating gonad ripening and spawning cycles in sea scallops on aquaculture farms and in the local wild populations. This is one of our lantern nets where we also keep scallops and where farmers grow them. We are working to see if there are differences in the timing and magnitude of reproduction or spawning in scallops grown in lantern nets versus scallops that are found in the wild population. Here you can see a female gonad, which is red, and a male gonad, which is white. So we collect males and females each week, July through September, on our farm and 2 other partner farms to dissect them to measure their gonads. This tells us how close they are to spawning. We also use an instrument called a sonde to conduct a vertical profile of water quality on the farms. This instrument collects pH, dissolved oxygen, chlorophyll, turbidity, and salinity data throughout the water column. You can also see Carl collecting a plankton sample in the background. Maddie's going to tell us about another piece of data that we collect.
This is a Niskin bottle. It's a way to collect water samples. We can prop it open, and then when I send down this messenger weight, it will hit a trigger and the ends will both snap shut, and this lets us collect water samples at a specific depth. So right now we're doing it right at our site, and we're just trying to collect water from right near our nets, and we're going to analyze it this winter, looking for scallop eggs mostly.
This data is also part of an environmental DNA, or eDNA, project with the University of Maine to detect scallop spawning events. So once we've collected the males and females from the nets and all of our farms, we bring them back to the island to dissect them. From each scallop, we measure the shell height, the total viscera weight, viscera are guts, the gonad weight, the meat weight, and the shell weight. In this video, you can see the scallop that has been dissected. The red part is the female gonad, which holds the eggs, and that big kind of tannish is the meat. As I mentioned, we work with other farms around Penobscot Bay as part of this project. One of them is Penn Bay Farm Scallops up in Stonington, Maine, owned by Marsden and Bobby Brewer. We visit their farm each week to collect our water samples and plankton samples and to take our scallops from the nets hanging on their farm. They not only give us scallops, but the farmers we work with help us identify research questions, like the one we're evaluating, and also share their knowledge about scallop farming. They're invaluable collaborators and have been fishing and farming along the coast of Maine for generations. They're scientists in their own right, and we could not do our work without them. Each week, we also collect scallops from the wild population to monitor GSIs to compare to our aquaculture scallops. This is a video of a scallop in its natural habitat on the bottom of the ocean. They like to hang out in cold, deep water on sandy and or shell-hash substrates. We collect them using scuba, and we also conduct population surveys of the local populations while we're at it. In this video, you can see us moving along the transect line where we collect data on scallop size and abundance, associated flora and fauna, and also the substrate that they're on. The data we collect from wild and aquaculture populations is helping us understand differences in biology between them, and also how reproduction on aquaculture farms may be contributing larvae to wild populations. We also partner with commercial harvesters and the Department of Marine Resources to get samples from commercial scallop vessels and their annual surveys. Farmers, fishermen, and managers are essential partners to conducting our work and making sure our research will be useful to scallop industries in Maine.
Great. Pepperwood Preserve encompasses 3,200 acres in the Mayacamas Mountains north of San Francisco in California, within the traditional homeland of the Wrapup people. We respect and honor past, present, and future generations of Wrapup and their continued connection to this land, and are grateful for the opportunity to gather in such a beautiful place and give their respect to its first inhabitants. Pepperwood's mission is to inspire conservation through science. Dr. Lisa McKaylee is the president and CEO at Pepperwood, where she has served for over 11 years. She believes that you can't understand nature without exploring it directly, and that together we are resilient. She is joined today by Carlos Diaz, principal engineer with Sonoma Water for 3 years. He likes applying cutting-edge science and research collaborations with local and regional partners to gather data to better manage water resources now and for the future with a changing climate. They will speak to us about providing real-time fire, flood, and drought hazard data.
Welcome to Pepperwood Preserve for Live from the Field. My name is Lisa McKaylee, and I'm the president and CEO at Pepperwood. And today I'm delighted to introduce my colleague Carlos Diaz from Sonoma Water, who will be part of our conversation today about how we are partnering with water agencies to share climate resilience data and to help develop real-time warning systems. Carlos, could you tell a little bit about yourself and your background?
Sure. My name is Carlos Diaz. I'm a principal engineer, civil engineer in California. Also consider myself an environmental hydrologist. I'm a principal engineer overseeing the design engineering group at Sonoma Water, but on the heels of the 2017 fires was really tasked with advancing a lot of gauging systems and trying to collect the real-time monitoring that was needed just to better understand conditions out in the field. And Lisa and Pepperwood Preserve have been an incredible partner in facilitating us deploying our systems. Some of my biggest inspirations have been some of my professors along the way. So I became passionate about water resources as an undergraduate up at Humboldt State University, and continued my pursuit of better understanding water resources, planning and management with a master's at Stanford University. I have a background in private consulting, dealing with not only infrastructure, but also really delving into hydrology, hydrologic and hydraulic modeling, and flood management. And so that was kind of my background before I came to Sonoma Water. At Sonoma Water, it's a little bit of everything. We provide water supply to 600,000 people here in Sonoma County. We also manage 10 sanitation districts spread out throughout the county, and we're also responsible for flood management. So it's really been a culmination of a lot of my life's pursuits and passions.
Yeah. And I have a lot of interests shared with Carlos. I am a river scientist and a watershed scientist. My background was more in physical sciences. One of my biggest inspirations was a oceanographer, geologist, Susan Humphreys, when I got to work on an oceanographic research vessel, who really got me excited about earth sciences and just studying how our earth evolves over time. I came to Pepperwood with a strong interest in downscaling climate futures. We had a partnership with Sonoma Water. I was just telling Carlos, our very first grant actually was to help develop future climate projections to inform water supply and habitat restoration, other kinds of work. So today we're going to talk a little bit about that history, and we're excited to share what we're doing with you. So Carlos and I are standing in front of the very first weather station with soil moisture monitoring that we installed at Pepperwood in 2010. And it's part of our Sentinel site network, where we use the preserve as a living laboratory to understand our key scientific question of how is climate changing both over time and space in our landscape. We have over 17 full-blown weather stations with soil moisture probes on the preserve now. And we also have micromet stations at 54 different long-term forest monitoring plots. But interestingly, when we started collecting this data, we didn't realize how valuable it would be to our partners at Sonoma Water. And so Carlos, could you talk a little bit about the value of this kind of soil moisture data, especially when we're dealing with climate hazards like droughts?
Yeah, absolutely. So, as Lisa mentioned, they're really pioneers in terms of, starting to collect the real valuable data. needed for, you know, water resources planners and managers to better manage our water resources. And soil moisture is really a critical component of that. Lisa had mentioned some of the climate change and just understanding how our watersheds and hydrological cycle is changing in both time and space, and collecting this data is really critical to establishing, you know, our conceptual framework for how our, you know, hydrological systems and ecosystems are reacting to a changing climate. And we're really fortunate for having partners like Pepperwood with the foresight to begin collecting this data and begin establishing these long-term data sets.
So could you provide an example of how soil moisture data might inform how you manage the reservoirs that are part of our water system, or perhaps the Russian River, which is really our main water source for over 600,000 people?
Yeah, absolutely. So soil moisture is really critical in understanding how, you know, any rainfall event is gonna be divvied up in between recharging, you know, our lands or turning into runoff. And so it's really critical to not only understanding flood response, so, you know, understanding what soil saturation levels are prior to a rainfall event gives us really good insight into how we might anticipate that rainfall turning into a flood event. If we have saturated soils, you know, nearly 100% of that rainfall is transformed into runoff. And on the flip side, it's also really critical to understanding drought. This year is a really great example of that, where we've just had such dispersed rainfall events of, you know, only half an inch, 3/4 of an inch at a time, and the soil moisture has been so depleted that, you know, most of that rainfall ends up recharging the soils and groundwater and really turning into very little runoff. So we haven't had the response in our reservoirs that we would have anticipated in the winter due to soil moisture conditions. And so again, collecting this data allows us to better understand the dynamics of how our reservoirs function and the long-term reliability of our water supply.
Carlos, I'm so excited that you're going to be able to explain what this brown tube is sitting here at Pepperwood as part of one rain system.
Yeah, so this is one of our rain gauge sites, one of the 22 sites I mentioned earlier that were installed around the Tubbs and Nuns fire footprints. This here is just a standpipe. The top section of the standpipe here contains a tipping bucket. That's how we measure rain in time. And we've also got a solar panel to recharge the battery and an antenna that beams this data in real time to Mount St. Helena, where we have a repeater, and that repeater bounces the data back to the water agency headquarters. And so what we have here in the standpipe is the actual transmitter itself.
Wow.
And so it's a lot of wires, but basically all we've got here is the battery I mentioned contained within this unit, solar power to charge the battery. We have a GPS that allows all the gauges to be on real time. That's really critical, so that every gauge knows exactly when to be transmitting data and they're not stepping on top of each other.
So the GPS is a global positioning system that's talking to a satellite?
Talking to a satellite and making sure that all the gauges are on the exact same time.
I guess the other partnership we should talk about, Carlos, is just the fire camera network. We've talked a lot about water today, but could you just share a little bit about, along with our emergency awareness around floods, how we partnered around the fire camera network?
Yeah, so the fire cameras was another initiative that grew out of the 2017 fires, and just the need to identify these fires before they became you know, so large that there's really nothing you can do about them. And so Sonoma Water initially funded 8 cameras. That was the genesis of the North Bay fire system.
And we were the first 2.
Yeah, and Pepperwood were the first 2 cameras.
And not only do the agencies look at the data, but all of our local communities now have real-time information on location of fire and fire spread. And that was really a game changer when fire came to our property again in 2019, that CAL FIRE knew where to hold the line. They actually came and fought the fire here at Pepperwood. And in 2019, the fire was prevented from getting into our urban areas. So Carlos, it's been so rewarding for us at Pepperwood to be in this relationship with Sonoma Water and to see our data getting used for really super important climate resilience challenges and benefiting everyone in our community.
Yeah, I think one of the benefits of the fires was just kind of convening all sorts of different organizations and agencies and groups and nonprofits in the county, and convening at the table and determining, what the questions were, what the problems were, and how we were going to go about answering those, fostering those relationships long term, in order to collect the data necessary. It not only benefits us in terms of managing our water resources, planning and management, and helping us answer questions that we have in order for us to do a better job, but that data is also available to countless researchers who may be advancing science and our understanding of the dynamics of drought, fire, and flood.
Great. The University of Akron Field Station is a living laboratory for the advancement of knowledge through ecological research, education, and stewardship of the natural world. It covers 540 acres on 3 different sites in Northeast Ohio and wants to make visible that these lands are the ancestral lands of Native people. Houette and Elena Statu are both PhD students of integrated bioscience at the university, where Thibaut also serves as a teaching assistant and Elena is a biomimicry fellow. They have been working at the field station for 2 years and like getting back out and appreciating nature, studying organisms in their environment, and sharing their research and thoughts with the public. They will tell us about their research projects studying root systems for bio-inspired design of building foundations and coastal infrastructure.
Hi, I'm Elena and this is Thibaut, and we're both Integrated Biosciences PhD students in Dr. Petra Gruber's Biodesign Lab at the University of Akron. And today we're here at our university field station, which is in Bath Nature Preserve, a local nature preserve for the community, and it's only about 20 minutes away from campus for us.
Hey, so Thibaut here. I'm a French architect, and in addition to the PhD, I'm also a teaching assistant for natural science biology labs at the University of Akron. For the PhD, I'm studying biomimicry for architecture. And just in a few words, biomimicry is basically the transfer of biological organism strategies to technical systems. So we're looking at how different organisms are going to perform different functions and adapt to environments, so we can abstract those strategies and transfer them to our technical systems towards sustainability. And for the PhD, I'm studying mainly 2 different topics. The first one is growing fungal mycelium on agricultural waste in order to make sustainable building materials. And the second one that we're going to talk about today is looking at the morphology and growth of root systems in order to design better foundation systems.
Hey, it's Elena again. So I'm a local engineer from the Midwest. I grew up both in Michigan and Ohio. And also, while I'm doing my PhD, I'm a biomimicry fellow with 3 organizations, Biohabitats, Cleveland Water Alliance, and ODNR Office of Coastal Management. So I work part-time with them on design and engineering projects, bringing a biomimicry lens to that work. And I also get to do some field work also, which is pretty cool. So I study for my PhD biomimicry for coastal engineering, specifically for the Great Lakes. And I look at tree species that exist along rivers and coastlines, specifically their root systems and how they stabilize the shoreline and protect the shoreline.
One issue is that there is currently a lack of consistent biological root trait data in the literature. So it's difficult to compare results between studies and find trends. So we're working on a technique to get 3D models of root systems to have this kind of automated process to analyze biological root traits. And to get the 3D models, we're using photogrammetry, which is basically taking a lot of pictures from different angles of one object, in this case the roots, and then we upload those pictures to a software that will reconstruct the 3D model virtually, and then we can analyze this 3D model. And to use this technique, we really need to increase the accuracy to get consistent data from the root models, and also increase the accessibility so we can use it in the field.
So we want the most accurate 3D models of these root systems so we can get things like root branching angle, root length, and root diameter to inform the designs of our different engineering applications. So first in the field, what we'll do is we'll measure all around the root wad, root length, root diameter, so we can compare these in-field measurements to the measurements we get on the computer with the reconstructed 3D model. And you can see these different treatments here where we're trying to improve the reconstruction. So there are markers, colored markers on the roots. We've got powder, which is just flour from the grocery store to really enhance the object in the photos. Today is a really cloudy day, which is great, so we get very minimal shadows on the root system when we're taking the photos. So we'll take the same 3D model across different treatments and compare those in-field measurements to the computer measurements. And with this process, we're working with Claudia. She's an undergrad biology student, and she's helping us with this comparison process.
So we're also working on the accessibility part of this technique, because the goal is that anybody around the world with their own imaging devices could come to a root system, and they would take pictures following a specific procedure, and then they could upload those pictures to a database where we could reconstruct the 3D model. But it's pretty difficult to actually image complex systems in the wild. So it was really great to start at our local University of Akron field station, where we could actually experiment with the technique, for example, using a phone, a digital camera, or a drone, testing also different marking techniques, and use the space and tools that we have here at the field station.
So for this project, talking to experts has been really helpful, both in root biology and in photogrammetry, especially to help optimize this technique. And while the study is still ongoing, we've learned some key findings. One is that the number of pictures taken, as well as the addition of powder specifically and markers, seem to help with the accuracy of the reconstruction of the 3D model. And the type of camera used does seem to make a difference, but we've noticed phones that have really good quality cameras give us really good 3D models, which is helpful for the citizen science process.
So this has not been a linear but an iterative process. So for the last 2 years, we've been optimizing these techniques, like getting slightly better each time. And we still are getting some mixed results between different experimental groups because our technique is slightly affected by the weather, for example, shadows, or even like the wind moving some parts of the root system. And in some cases, we also cannot access all around the root system to take the pictures that we want. But ultimately, the results of this technique will actually lead to more sustainable bio-inspired design for building foundations and also coastal infrastructures. In the case of building foundations, the goal is to actually adapt the morphology of the foundation system to different environments. So for example, in cities, we want less soil compaction to increase water infiltration and have less flooding. In environments like slopes, on slopes or coastlines, we actually want to prevent erosion. So the goal is to kind of integrate also multifunctionality, so we can also store resources and share them between buildings, but also with the environment.
And in the case of coastal infrastructure, for me, a lot of the practices used today are really old, traditional, and a lot of the infrastructure displaces natural habitat like oyster reefs and wetlands, which provide a lot of great ecosystem services. And something as simple as like a rock pile or concrete wall in front of someone's home can cause downstream erosion issues for the next door neighbor. That same concrete wall, if waves are crashing against it, can churn up the water, make it cloudy, so it's really hard for plants and fish to thrive. And so a lot of issues are caused by current infrastructure, which then is solved by even more engineering solutions, which completely changes our coastline. So what I'm hoping to do with the study of root systems is start to renaturalize our coastline by creating bio-inspired infrastructure that mimics nature in form, function, and material. And so what's really great about the field station, since it's open to the public, we can interact with people talking about our different research applications in the study of root systems. And in this video, we hope you've learned something from us today and about our research.
And let us know.
Hello. Thank you so much for those amazing presentations, everybody. We really got lucky with an amazing group of people here, and they are actually all here. I believe maybe Elena is not here to answer our questions again. So I just want to encourage everybody who has questions for them to type them into the Q&A, not the chat. That's just an easier way for us to organize them. So please do go ahead and type any questions that you have for the researchers into the chat now. And I'm going to get started with a couple others. So we do have college classes here that have a time constraint of 50 minutes. So we'll initially give priority to those questions. So if you're a student, make sure and type your name and the word student there, and we'll answer your questions first. Please make sure your full name is visible. The panelists are going to try and keep their answers to a minute or less so that we can get to as many of the great questions as possible. So if the panelists want to turn on their video and audio now, we'll get all started with one question that we'll ask to all of the panelists. And maybe we can start with Phebe Jekylek, move on to Lisa McKaylee and Carlos Diaz, and then finish up with Thibaut, and if Elena can join us as well. So first question, can you tell us or remind us what drew you to studying applied science questions in the first place?
Such a good question. So I live up here along the coast of Maine, in a town that has a very strong working waterfront. And I also work on an island off the coast that is part of the second largest lobster harbor in Maine. So I am exposed to the working waterfronts here, and you know, the culture in Maine is very immersed in the fishing industry. And I learned pretty early on after college kind of how important including and seeing fishermen as scientists was to the scientific process. So I would say right after college, one of my first jobs was as an observer. So you go out to sea with fishermen and you are recording biological data on the catch that they're catching. You know, you're recording bycatch data, all that kind of stuff. And you're living at sea with fishermen and really getting to see what they do. And, you know, that was my first exposure to learning from those guys, because they're on the water every single day. You know, they are making decisions in a very scientific way. And so it was really important for me to be doing work that would both incorporate that existing knowledge, that traditional ecological knowledge that they hold, and also work that would serve you know, some of their interests and some of the questions that they were bringing up about what's happening to the resources that they depend on in their lives. So it's, I think applied work is really important, and it's a really great way to work with communities, as some of, you know, as our other panelists have shown as well. And I think that's an important thing to remember when doing science. Science should impact community.
Did you want me to go next, Kerry?
That would be great. Thank you, Lisa.
Yeah, so I really think as soon as I realized that I loved science and was pretty good at math, that I wanted to figure out how to serve. I come from a family of people in service. So, and it really seemed like either the environmental crisis was really a great fit for bringing science to bear on serving people. My first job was at the Environmental Protection Agency as an entry-level scientist, so really trying to enforce the laws that benefit people. But I learned early on there that one of the things that really interests me about the environment is how everything is connected, and I love interdisciplinary research. And so I found water being this great connector, and I had a mentor in the water division at the EPA who was like, this might be the study topic. So I really kind of went to work trying to protect the environment with my undergrad degree, and then figured out a topic that was very applicable in terms of environmental protection. And then I went back to graduate school to get more advanced in that area. And I really wanted to work on river restoration. That's what drew me into this work. I wanted to work on turning things around after I worked at the EPA, where we were really good at stopping bad things from happening. But then I wanted to use my science degree for working on positive projects, positive change. So.
Right. Tough shoes to follow up there, Lisa. Hi, everyone. Carlos Diaz here. I'd say I share a lot in common with Lisa in terms of my background and really being exposed to natural treatment systems as an undergrad up at Humboldt State University. The city of Arcata has, a world-renowned natural wetlands complex that treats all of the city's wastewater, and it was really kind of understanding these natural systems that really drew me into applied science. Just really attracted by the complexity of natural systems, the biological mechanisms, mechanical, physical, chemical mechanisms, all kind of working together in this case to improve water quality, right? To protect the water resources of Humboldt Bay. So really diving into kind of understanding water quality and how it, impacted our community's environment really drew me in. And I'd say just in larger context, anything water resources really drew me in, understanding groundwater, kind of surface water interactions. As an undergraduate, that's what kind of drew me initially to a master's degree, where I was completely kind of went in another direction because I became fascinated by microbiology and more biogeochemical systems. Following, I guess, my undergraduate, really diving into my professional career, river restoration really drew me in, and just understanding kind of natural hydrological and geomorphic processes and how we could, understand kind of those processes and use that in the design of our projects, really to kind of benefit, not only us and our local communities, but the environment and the ecology and everything that we all depend on. So that's my response. Thibaut?
Yeah, thank you. And so for me, I'm coming from architecture, so it was pretty natural for me to look at applied science, looking at ways to use the biological research and biomimicry method to actually design more sustainable buildings. So partnering also with, the applied side, so that the designs that we think of are actually used and can change the ways that buildings are developed. And so we're looking at different scales, like for example, for individual components in the buildings, but also how we could cycle some of the resources more efficiently and consider the entire life cycle of buildings. So really trying to do the research to gather data, but also apply it. So it's making a change in how we design stuff. And also, like, by having this kind of citizen science part in the project, it's also able to invite the public to participate, introduce them to some of the research topics, and also, kind of show them what are some of the main questions that we're looking at, how they can actually help us, and how they can change some of the habits to help all of us going towards sustainability.
Well, it perfectly leads into one of the questions that somebody asked Thibaut. They asked, how would the citizen scientists be trained and what would they need to do specifically?
Okay, so we're looking at doing a kind of a manual where we could have a description of the steps that anybody could follow, and then they would just use whatever imaging device they have, and they could find a root system that is exposed and go to that root system, take pictures following, again, this kind of manual. And then they would be able to upload those pictures on a database where we could reconstruct 3D models. And then we could share with them the 3D models, but also like some of the data that we collect from those 3D models. So this is not done yet, but it's our hope to really like do this kind of manual and integrate the public to actually gather data from all over the world.
Fantastic. The next question is for Lisa and Carlos. What other measurements are you using to monitor streams at Pepperwood? Is there emerging tech?
Yes, there's so much cool ways we are measuring streams at Pepperwood. This is one of the great things about being a field station is we put in a basic stream gauge early on. Carlos's team put in more flood rain gauge warning systems, and there's additional warning flood warning gauges downstream. We now are putting up, we're pretty high up in the watershed, so we're putting in 2 monitoring weirs as part of a stream flow enhancement study that we're doing. And they are hard to build in a rough stream, but we'll be capturing both base flow and peak flow in there. And we've just gotten flux towers. So we're actually measuring evapotranspiration directly out of 2 different watersheds, one that's being thinned and one that's not. And you're usually estimating that. So it's very related to stream flow. We're measuring it directly. And we have about 20 soil moisture probes all over the preserve. You heard us talking about soil moisture. So we're really trying to get the whole water balance. We will be drilling into the bedrock to look at fractured bedrock water. And we're also experimenting now with the USGS National Innovation Center on drone flights and testing out sensors that you can put on a drone to look at soil moisture. Now this all, you asked about stream flow, but this all ties into stream flow because you need to really understand the whole water balance to get to stream flow. So those are all coming together and that data is shared. There's a question about data sharing here too. All of our data is shared through Sonoma Water through a public portal. And so that data is available to the water agency to steward the data, but it's also available to everyone in the public to know what's going on with their watershed.
Wonderful. We have another student question I'd like to get to right before we send students off. This one is from a student who says, are there any other places with opportunity to conduct research with scallops? I love them and want to know more.
Yes, there are many different kinds of scallops. You can find different species of scallops all over the world, and in both aquaculture and wild populations. Up here along the coast of Maine, we have sea scallops, but you find sea scallops all the way down to the mid-Atlantic as well. We also have bay scallops here in New England. Those are smaller. You find those from kind of Massachusetts and south. But there are all kinds of scallops and all kinds of people doing really amazing work, trying to understand a lot of aquaculture populations of scallops and wild populations, their biology, their responses to climate change because they are important commercial species. So there's all kinds of ways to get involved in scallop work, and feel free to reach out to me directly if you want to kind of learn more about what we do, or if you want to be connected with other people doing work. My email, I'll put my email in the chat for everybody.
It is amazing to me that we are already at 1:52 p.m. Pacific time, but I am going to let students that have to leave go, and we will continue the chat for another 10 minutes or so. So thank you everybody for joining us. We will be emailing follow-up information, and someone asked if this was recorded. It will be recorded and posted at the same place that you registered, and we'll make sure and send that link out in the follow-up email as well. Now, if you're an instructor, it would be fantastic if you could take a quick survey. I just put that link in the chat, and we will also email that out. So thank you to our speakers, Carlos Diaz, Thibaut Houette, Phebe Jekylek, Lisa McKaylee, and Elena Statue, who might actually still be joining in a minute. We also want to thank the Organization of Biological Field Stations, the Center for Environmental Inquiry at Sonoma State. And please do learn more about their series of virtual events at thevirtualfield.org and cei.sonoma.edu. Thank you also to the National Science Foundation for funding this project and all the wonderful people behind the scenes who made it possible. So in that email in the next few days, we'll send you a follow-up recording of this event, answers to questions we don't get to address today live, and a link to that survey. Now, we have one other live from the field event coming up on October 25th, and this one will be on freshwater invasions. So you can learn more about that one at cei.sonoma.edu/freshwater-invasions. And then please also check out the recorded events at thevirtualfield.org. And they're all available under Live from the Field page under virtual visits. There's also a website with the featured Cite today resources and information on publications from each researcher with educational tools. So I am going to go ahead and finish questions for those who can stay. So our next one comes from a student who asked Lisa and Carlos, what made you want to do this type of research on roots? But I guess she's actually talking to Thibaut and Elena because it says on roots.
Yeah, so I actually read the book called The Hidden Life of Trees, like before the PhD, and I was truly amazed by roots and all of the things they do that I had no idea could be done. And then when you compare to our engineered systems, especially for building foundations, which are like still pretty traditional and did not evolve that much over the last years with our like emerging technologies, we really thought that could be an opportunity to design more sustainable foundations, especially like when you look at cities and problems with flooding and like all of the kind of resources consumption and all of that in the building industry. We really tried to look at root systems as a way to design more sustainable foundations. So really like learning about root systems when I looked at this book, and you know, you start by seeing this kind of biological strategies that root systems have, and then you start to look at the scientific literature and you realize that there's so much more behind it, and you just delve into all of this and just inspires you a lot. But yeah, this book was really mind-blowing for me.
I've actually read that book as well. I love hearing the stories of how people got to where they got. So we have another student here who's asking, Pepperwood, are you working with anyone else to monitor wildlife response to changing water conditions?
Well, we're working on it really hard ourselves, actually. We have 50 wildlife cameras at 2 reserves, ours and one north of us, that have been running since almost 2012. So we have a really great ongoing data set of wildlife fluctuations over time, which is capturing the fires. We're really focusing on the fire impacts right now, but we will also be capturing the impacts on drought. And we have other wildlife indicators, including cover boards for lizards out in our forest plots and bird surveys that we do. So we're really interested in that question. We don't have answers for you right now. We certainly do share our data with we're working with the Bear Collaborative right now, and so that's a very big collaborative effort, and we're trying to get resources to combine our wildlife camera with other organizations, but we haven't been funded yet to do that. But we'd love to do more of a regional assessment with the many partners we work with who also are using wildlife camera data in particular.
Thank you, Lisa. This next one is for Phoebe. How can fishermen and aquaculturists use findings from your research?
Great question. So some of the work that we're interested in is investigating if there are differences in spawn timing, in spawning is reproduction, or the magnitude, like how much reproduction is happening in aquaculture and wild populations. And also understanding if by growing scallops in aquaculture, putting them in these very high densities on these farms, if we're actually increasing fertilization, resulting in more larval supply. So increasing more scallops, or, you know, are we increasing the number of scallops that are out there to be harvested back into aquaculture or to settle out into the wild population? So some of those questions have pretty big management implications when it comes to fisheries management and making decisions about perhaps where to site aquaculture farms, how you can best set up aquaculture farms to act as fertilization enhancement areas. So some of those issues are very, very of high interest to fishermen and aquaculturists, because all aquaculturists actually gather their baby scallops from the wild population. So they put out these spat bags. They're called spat bags. Baby scallops are called spat. So they put out these bags in the water column where little baby scallops actually settle in them. And so if they can understand maybe where scallops are coming from and where they could put them in relation to our farms, they might be able to just go ahead and catch the scallops from their farms that are creating new babies. So Yeah, there's lots of other things happening. There's climate change effects that we're trying to understand, comparing wild and aquaculture populations. Our scallops that are grown up in aquaculture, are they responding to changes in climate differently than those in the wild population? And also, what can we understand about the environment that these scallop populations are growing up in? Scallops that are grown in aquaculture are grown up in the water column. So they're grown about 30 or 40 feet above the bottom, but 20 feet below the surface. So they're kind of hanging in these nets vertically in the water column. Scallops in the wild, they grow up on the very bottom. So they settle on the bottom, on the sandy bottom, and then they just grow up there. So these are two very different environments. When you get out here along the coast of Maine, they could have different food availability. They can have different flow regimes. They can have different temperatures. So what do all those different changing environments do to biology, to the ecology of scallops? And then what does that mean for fisheries? So there's a lot more questions out there. And I'll tell you, literally every single time I talk to either a grower or a fisherman, there is a new question to be explored. So always talk to the people that are, have their hands dirty with what you're interested in studying, because they have so many good ideas and so many good questions. So I have no doubt that I'm going to have my hands full for a long time with more scallop questions.
All right, I'm going to try and wrap up with one more if we can squeeze it in. And this one is for Carlos and Lisa, and it's a complicated, multifaceted question. So I'm kind of try and bring it down a little bit. And it's about how understanding the data on drought conditions can convert into planning water use, into who actually is deciding what to do with this information that you're producing, and how those decisions are made, and how Pepperwood is involved with those decisions.
I feel like I should let Carlos lead, because this is a
I'm happy to
complicated
take that,
question,
Lisa.
and it's a scientific and a political question too. So
Yes, it is. Let's see. I think that's at the heart of water scarcity is kind of competing for limited available resources, right? And understanding the surface water groundwater connection is, I think, really critical to understanding where is the balance and how do we strike that balance, and certainly maintaining minimum in-stream flows for fish is a very hot topic these days. I mean, it's gone all the way up to the State Water Resources Control Board and the Division of Water Rights, and a lot of players getting involved there. Marquess Creek, where Pepperwood is situated, is one of kind of 5 key streams in California that have been identified for better understanding these surface water groundwater connections and interactions and the water balance as a whole, so we can understand what policies need to be made in better managing our limited water resources, so that we can continue to support our local economy and the structure of that economy, but also supporting our local ecology and meeting and hopefully exceeding those minimum kind of needs of fish. And certainly a very hot topic these days in the drought, with I think some pretty heavy hands coming down. Well, some might consider them heavy, some might consider them not heavy enough, but curtailing kind of water rights users. So we've reached that in the Russian River Basin this year, where there's simply not enough water for the landscape and human needs and ecological needs, and so there have begun to be curtailments kind of limiting what water users can extract from the system. I think all this points to the need for more groundwater recharge, and really kind of making better use of water when it's available, so that it's plentiful and around when it's not available.
And one of the things about where we live is that our water all comes as rainfall. We don't have snowpack like many other parts of the West. And so we think of Pepperwood as a catchment for the watersheds downstream, and demonstrating how to take care of that catchment. Our preserve generates about 50,000 acre feet of water a year for downstream users. And what we think is key for resilience is helping to restore kind of the sponginess of the watershed and increasing the water holding capacity at Pepperwood that probably has been degraded from past agricultural practices, et cetera. So adding carbon to the ground sequesters carbon from the atmosphere, but it also helps hold more water in place that then releases it more gently. And that's why we look at the whole water balance. I mean, really the water that comes out in streams is the water that was stored underground. So as Carlos says, the better we understand those systems, the more accurately the water agency knows how much water there is. And then it's always a social and political process to talk about how we manage that resource collectively. And it's a whole nother, I mean, water rights are different based on every kind of water you have, but managing our underground water and our aquifers is really, that's our savings account. That's our backup account. And so that's why there's a lot of movement now about connecting these dots, and our data is really providing critical understanding of the relationship between what falls as rain and then what we get out of the streams.
I think that's a perfect way to describe this event in total, right? Is that connection to the research that's going on at these places and everybody who's attending this event and is in our community. So thank you all for articulating that and making that connection so clear for everybody today. I'm going to have to finish up the questions by email because we are over time. So I just want to say thank you one more time to all of our presenters. Somebody was asking about getting more information about you all. So I'm going to send out a resources document, and that has publications that each of these researchers has put out about the work they discussed and some other general information. And I'll also have a link to each institution's website so that you can learn more that way as well. So with that, I am going to ask all of our panelists to just wave goodbye and thank everybody so much for coming. Hope to see you again soon in the field. Take care, everybody.